Thank you very much for the invitation. This is going to be a bit of a challenge to describe how a physicist can work on cancer and explain it to a multidisciplinary audience. So let's see how it goes. Anyway, as Jose mentioned, I at Rice University and brought here with the help of CIPRIT 3 years ago, and we run an NSF center on applying biological physics to many, many different problems in the living world. And over the last few years, we've been thinking very much about how to bring that expertise to cancer research. Now, when you think about the role of physics in cancer, this is not all that an unusual question because there are many aspects in which physics has and will continue to impact cancer research. And all of you, if you thought about that, would come up with several immediate examples. You would think about new experimental methods. You know, MRI is not new anymore, but continues to be improved as a modality for imaging cancer. Enhanced treatments. There's actually an article today in CNN on proton therapy for cancer as a new way to bring physics ideas of interaction of particles with cells to perhaps treat otherwise untreatable cancers by standard radiation methods. There are nanoparticle applications. There are sophisticated data analysis tools that we've actually seen some of when you hear talks about genomes. A lot of those methods have been developed by people coming from physical science who are more experienced and have more history of dealing with large data sets in the context of physical measurements. But today I'm actually going to talk about yet a different direction of physicists thinking about cancer. The different direction is making use of the fact that the physics community, the theoretical physics community, has methods and ideas about how to look at complex systems ranging from the climate to complex engineered systems to now biology, where we can use mathematical methods, computer simulations, other techniques to try to extract core mechanistic ideas and thereby somehow capture the essence of a complex phenomena and therefore understand it better and, of course, therefore manipulate it better. Now, it may sound like a crazy idea to think that physicists are going to be able to do that in something as complicated as cancer. So the easiest way to at least provide motivation for the work is to quote one of the leading cancer biologists, Bob Weinberg, in his two seminal papers, Hallmarks of Cancer and then Hallmarks of Cancer Revisited, where at the end of those papers, after discussing the conceptual foundations that many of us use for thinking about cancer and the changes that bring cells from normal cells to cancer cells, he indicated that he was longing for a day in which cancer research could have a sort of logical consistency and mathematical foundation and being based on principles that rivaled chemistry and physics. And so you can read his words for yourself, and I think of that as very strong motivation to at least try to do that, to take ideas that are commonplace in physics applied to other complex systems and ask what happens if you try to apply them to cancer? Where do you get? What can you do? How can you contribute to the field? So I'm not alone in doing this. There's now a community of people indicated by covers of Nature, special issues, covers of physics journals, Physics World, showing that physicists are trying to do this in a variety of ways. And I want to give you a snapshot of one set of issues that we've looked at. The snapshot is, of course, has technical details, but the talk really, I want to transcend the technical details and just give you an idea of how the thought process works and how we think this thought process can augment some of the more traditional ideas of how to proceed to understand aspects of cancer progression and cancer initiation. So here is, as you've already seen today in a variety of contexts, the Hallmarks of Cancer. This is the original version, and this is a series of changes that cancer cells have to undergo in order to eventually become full-fledged cancer and eventually cause metastatic disease. And each of those can be studied from a variety of perspectives. And I'm going to focus on one aspect. This is the aspect listed here as tissue invasion and metastases. As we heard already today, metastatic disease is what kills most patients. If the disease is localized, if you diagnose the tumor when it still has not metastasized significantly, you can typically get good survival rates from surgery and from localized radiation therapy. So this is really the, from a curative perspective, this is really the challenge. And I'm going to talk about how thinking about this from a more physical science perspective can perhaps give some new insights into what's going on. And then you can judge for yourself if this is a fruitful way to address some of the questions that arise in other aspects of cancer, and whether this is a sort of interesting possibility for how to reorganize our approaches to cancer to make them even more interdisciplinary than they are today. Okay, so here goes. So when you think about the metastatic cascade, the way in which tumor cells initially go from being uncontrolled growing cells in a primary tumor, as indicated in this picture by this sort of green mass of cells, to really being a danger to the health of the patient, which is indicated by those cells leaving the primary tumor, making it through the blood system, eventually emerging in other organs of the body and establishing secondary tumors. When you think about that process, there's sort of a question about how cells can manage to do that. We all know that cells, when they're staying put and growing, have certain properties which are called epithelial properties. They stick very strongly to each other. This is why your skin forms a good protective boundary. They don't move. You're not interested in having your organ cells spontaneously pick up and move to other parts of the body. So they exhibit properties. Properties, of course, are controlled by gene expression, which are natural for behavior of normal tissue, normal epithelial-like tissues. In order for those cells to leave the primary tumor and eventually establish secondary tumors elsewhere in the body, they have to change those characteristics. They have to change their phenotype. And one of the things they need to establish is cells have to undergo a phenotypic change. They have to change from having properties that are characteristic of epithelial cells. They have to change to having properties of motility, to being less sticky to each other, to being able to transit through the blood system. And then the opposite has to happen, because they have to establish a secondary tumor in another tissue. So they have to go to another part of the body, and in that other part of the body, they have to stop moving, settle down, attach to the local environment, and of course then start growing again as a secondary tumor. So there's a series of very specific types of changes that need to take place in order for cells to go through this metastatic cascade. In the cancer community, these changes are collectively called the epithelial-mesenchymal transition or transformation, and the reverse, going backwards, is typically called the mesenchymal to epithelial transformation. So when a physicist, so this is a cartoon picture. This is from a review article published last year in Nature Medicine. And this review article is a great cartoon of what's going on, but doesn't provide any quantitative insights into knowing how do cells do this, how often do cells do this, how can one change the rates in which cells do this, how many cells survive this transition. There are many quantitative questions that arise when one looks at a picture like this from the eyes of somebody who is a theoretical physicist, that we want not just a descriptive version of what's going on, we want to transform that into a quantitative description of what's going on. So how do we do that? Well, we focus on one aspect of the problem. So I already said that we were interested in this change of phenotypic behavior, a phenotypic transition from one type to the other. Now, how do cells do that? Well, cells have networks of genes, and those networks of genes are coupled to each other, and the net effect is that cells are miniature information processing systems. They take in input from their environment, from other cells, including the immune cells that we heard about in the last talk, and based on those inputs, they use their genetic networks in the same sense that an electronic system would use an electronic circuit to then decide what genes they're going to express. Are they going to be motile? Are they going to stick to each other? Are they going to try to transit to another part of the body? Those networks of genes, we've understood quite a lot of them over the years, starting from work on very fundamental biological systems, such as bacteria, simple eukaryotes such as yeast, going on towards mammalian cells, which are of course more complex, but obey many of the same general principles. So these interactions between these genes, where one gene affects the transcription of another gene, affects the translation of another gene's RNA into proteins, form this computational network that then allows the cell to respond to external environments. Now, if the cell were behaving normally, if the cell were a normal epithelial cell, then of course its proper response to those environmental signals would be to stay put, to not do anything, to grow only under extraordinary circumstances such as when a wound occurred. However, the cancer cells, because of their mutational changes, are responding improperly. They're using those same networks, they're making the same decisions, but they're actually making them incorrectly. So the mutations in this language are essentially misguided information pathways which tell the cell to behave in a way which is perfectly natural under a different time, different signals, different place, which then, in the context of cancer, leads to aberrant, deadly behavior. So the biology community, the cancer community, of course, spends many years understanding the details of how those circuits work. Here is just an example of a type of paper that one can find many of in the cancer literature. I'm not going to go through this. This is obviously not my work. But what the cancer community has done is identified some of the key elements of these computational circuits. In this case, there are proteins called ZEB. There's RNA molecules called miR200, which collectively control how these circuits work and try to offer some insight into how the cells are constructed in order to make these decisions. And of course, understanding mutations in these genes and these elements then offer insight into how cells, when they become malignant, are making the wrong decisions. So one can take data like this, not one paper, but let's say hundreds of papers, and distill it down to trying to understand what are these circuits that control what cells are doing. So this is a cell circuit diagram. It's just a simplified version, which tries to explain how the cell makes a decision between staying put, remaining epithelial, or moving, which in this case means starting the metastatic cascade. And I just want to point out a few features here. What you have here is you have a computational network. You have a structured network. This is an equivalent for a cell of what it would be if I would show you a circuit diagram for the inside of some electronic component. The idea is you have elements that mutually affect each other together with inputs and outputs for the circuit. And one key ingredient of a circuit that's making a decision like this is the idea of two parts of the system that are mutually inhibiting each other. So let me show you an example. This is a protein. It happens to have the name snail for historical reasons in the cancer community. This protein, snail, when it's large, when its concentration is high, gives rise to motility behavior. The cells begin to move. This is another element, an element which is a small RNA molecule, microRNA 34, and its related family of microRNA 34 molecules, which has the property that when it's high, the cell, all the outputs of this circuit convince the cell to stay put. And the relationship between the snail protein and the microRNA 34 is mutual antagonism. The microRNA tries to suppress the snail tries to suppress the microRNA 34. In a given cell, one of them wins that battle. It depends on the inputs. Of course, you can bias the cell one way or the other. It depends on other components of the circuit. And at the end of the day, the cell picks one of those two choices. The cell has multiple possible states, and the inputs drive the cell towards making a decision about how it's going to behave phenotypically. Now, a circuit diagram, from a point of view of physics, is a diagram of the connections. But as you know, if any of you, many of you, I think, are from engineering or more orientations in physics, it's not just enough to draw the circuit. You actually need device characteristics. You need to know how the concentration, in this case of this protein, quantitatively affects the concentration of this microRNA. And conversely, you need to know how the concentration of the input quantitatively affects one of the inputs into this circuit. So we developed a formalism that allows one to do that. I'm not going to describe it in detail. It's essentially a way of saying if we knew, for example, the concentration of some microRNA. MicroRNAs are small RNA molecules that interfere with protein production. If we know that concentration and we know how many ways in which that microRNA molecule can attach to the messenger RNA, that's the number of binding sites in the messenger RNA tail that is responsive to that microRNA, we can quantitatively predict how much will be suppressed by that system. So we go through creating a quantitative model based on the biological data, and then we try to understand, using that model, how the system behaves in response to various external stimuli. So for example, an external stimulus that could be responsible for a cell trying to make the transition from non-motile, epithelial-like to motile, which is called mesenchymal-like, would be something that externally increases the concentration of the snail protein. We just heard about an example of such a signal. There's a signal called TGF-beta, which can be released by various cells in the environment, and that signal will take cells and push them along the direction of becoming motile. So if that signal is being released for various reasons, that will increase the chance of cells undergoing this transition to being metastatically competent. And when we study, for example, the circuit that we derive from this, what we discovered is something that was maybe not surprising, but at least somewhat interesting. What we discovered is that cells actually can have multiple fates. We started out by assuming or thinking that cells had two choices. They could either be motile or non-motile. It turns out cells can also have intermediate fates. They can have some range of properties where they're partially motile, but they're still adherent to each other. They still stick to each other. Now, what does the consequence of that? So, for example, before talking about the immediate consequence of that, so the first thing you want to do when you go through the trouble of creating such a theoretical framework for asking a question and coming up with a tentative answer to that question is you want to go and see if the data out there supports this new type of possible phenotype, a hybrid phenotype, where cells are both motile but still retain some of the classical properties of epithelial cells, in particular that they stick to each other. So it turns out that one of the groups at MD Anderson, the Semhanash, had been doing proteomic studies of a variety of lung cancer cell lines, and independently of our work, which was being done at a sort of more general level, he looked at whether his cells could be classified as being epithelial-like, non-motile, mesenchymal-like, which means motile, and when he tried to classify cells in those two types, he discovered that that worked for roughly half of his 40-odd cell lines, and then a large number of other cell lines resisted a simple classification. They seemed to have mixed properties, and actually, when we compare in some detail with what the data looks like, this is now proteomics data for these lung cancer cell lines, compared to what we would predict based on our now multiple state model. Actually, it offers good insight, and this needs to be worked on more, but it offers new insight into how you actually get cell lines and cells in general, which do not obey this simple classification. It gives you new insight, for example, into what you should be doing when you try to classify cells by proteomic data, which is one of the directions which is quite interesting for trying to make prognostic predictions of metastatic capability. From the point of view of function of the cells, I was stressing genes, and this gene turns on and that gene turns on, but cells use genes to make decisions about function, about behavior. So let me show you now the consequences in a simple assay initially of what it means to say a cell goes into a purely motile state or a cell goes into only a hybrid, partially motile state. So this is now experiments done by collaborators in Tel Aviv. It's a mouse cell line. And on the left-hand side is what happens if you have cells that move where they have been convinced by external signals, in this case the chemical TGF-beta, to become completely motile. So here is, just watch the experiment, you'll see what I mean. The cells move and they break free of the bottom tissue. So cells move, and then what happens is cells leave. So if you think of this as being a laboratory analog of cells that are now becoming motile and leaving the primary tumor, what you see here is the leaving the primary tumor is like boiling off of individual cells. They all move and they boil off and they proceed to try to spread throughout the system as individual single cells. On the other hand, you have an intermediate choice. This is now the same thing where the cells have been convinced to move, but these cells now have this hybrid property. These have adhesion with motility, and what happens instead is the cells move. There's now a progression, but the cells move as clusters. They move coherently. And once we have a theory, once we have an understanding of how the genes control the biomolecules which create these possibilities, then the thing we can do is we can verify our intuition by creating simple, simulation models, what's going on. This is now the computer version of that. So here is, again, cells now in the computer which boil off. These are cells that our equations for those cells are motility, but no adhesion to each other. And conversely, if the cells have adhesion proteins, even though they become motile, then you get cells move, but they remain attached to each other. They remain collective. So what we can do is we can, first of all, understand that transition to motility is not an all or nothing affair. You can become completely motile, fully escape, single cell individual behavior, but there's also an intermediate state. The intermediate state, the cells still move. They move attached to each other in clusters, and the cells retain properties of both the new state, well, the old state, the purely epithelial state, while picking up properties that enable them to move. Now, one of the things we can do, I want to just spend a few minutes talking about extensions of this, and I'll come back to why that's interesting and important. But one of the things that's always struck me is that there's relationships between this motility transformation, this EMT transformation, and properties of cells that to me didn't seem, when I first started learning about the subject, didn't seem to me completely connected. So for example, there's a notion stressed in a number of seminal papers by Sandori Mani at MD Anderson and Bob Weinberg of connections between the notion of cells becoming metastatically competent, leaving the primary tumor, and becoming more like stem cells, and particularly more like stem cells, the most important consequence of that is becoming much more drug resistant. So there are many experiments that sort of look like this. This is just one example, looking at chemotherapy resistant for cells, and this is in the context of prostate cancer, and showing that when cells undergo transitions to becoming motile, as they go through this epithelial to mesenchymal transition, not only do they become motile, but they also pick up properties of drug resistance due to a variety of mechanisms. I don't want to talk about the details of how this occurs mechanistically, but the point is, from the modeling perspective, from the theoretical perspective, what we're asking is, we have a module, a computational module, a decision-making module within the cell that decides, and that's the top one, that decides based on the inputs and based on the mutational profile of the cells if the cell is going to become motile or not. There's another module with different genes, different interactions, that decides based on inputs and based on mutational profiles if the cell is going to exhibit properties we associate with stemness, including enhanced drug resistance, including tumor initiation in proper assays. And then we can ask the question of how are these things coupled to each other? How correlated are the properties that we associate with motility and the properties that we associate with stemness or cancer stem cells? And again, this is something we've studied. We've studied this together with Sandori Mani of MD Anderson, and we've shown that the answer is it depends. In some backgrounds, in some cell lines, in some tumors, these things are very highly correlated with each other. your motility state directly determines whether you'll be stem-like or not. In other backgrounds, the connection is less strong, and you can have a variety of possible behaviors. And we can go a long way towards making sense of what is a rather confusing literature at the moment, various people observing that in some cases, cells become more stem-like when they become more motile. In other cases, they become more stem-like when they're less motile. This type of analysis can basically try to create a path through the fog of competing experiments on competing tumors, on competing cell lines. So I want to finish with two things. First of all, why is this important? Why do we care? Why have I emphasized this particular aspect of which we've tried to create a new path towards understanding by using mathematical models and biological physics? Well, it turns out, and this has now been shown in some very recent papers, it turns out that not all metastatic cells are created equal in terms of their capability to actually go and colonize distant regions and create secondary tumors. The logic is that I told you about the first half of the sort of conundrum of the cells. They leave the primary tumor, so they have to pick up these motility phenotypes. But then I also mentioned that they have to then revert. They have to go backwards. And reverting means they have to then somehow forget these motility phenotypes and revert to being dominated by growth, by just staying in place, sticking to their neighbors, and just growing and establishing a secondary tumor. So it turns out that in recent work published in Cell, that the cells that are most capable of doing that are exactly the cells that seem to be in this hybrid state. So why is that? Well, cells that are in this hybrid state, this was shown by showing that cells that go through the blood system as clusters of cells, cells that metastases that were created by cells not moving individually to the blood, but by moving collectively as clusters that were adherent to each other, those were, in this particular assay, 50 times more likely to be able to establish a secondary growth than cells were if they went through the system individually. And from the point of view of our analysis, it's actually somewhat logical. The cell has to go backwards in this transition. And it's a lot easier to go backwards if you've only gone partially forward. So our cells, our state, which is a partial state, has these residual properties that are still partially epithelial, partially mesenchymal, is much easier to revert back to purely epithelial and therefore start the growth process again than it is if a cell goes completely over to the mesenchymal state. And so it's not surprising then that cells that manage to transit get into the blood system, and the signature of that transit as a partial state would be that it occurs as a cluster of cells, that cells that manage to do that, when they land in the secondary spot, are much more likely to successfully, from their perspectively, very unsuccessfully from the patient's perspective, establish a secondary tumor in that spot. So the natural consequence of proceeding down this line of thought is that right now we sort of look at various genes for signatures, for prognosis, for how one might decide what are the chances of, let's say in prostate cancer, of a localized tumor becoming metastatic as a way to figure out what the treatment modalities might be. And if one has a more sophisticated understanding of what cells really are the ones that are the critical ones, which are cells that are interacting with each other, they're still adherent, they're able to establish new colonies, they're resistant to drugs therapy. I didn't show the picture, but there's evidence that cells in these hybrid states are also more resistant to immune therapies. They upregulate these regulatory T cell parts of their system. That could offer both a prognostic advantage of trying to figure out how we could better predict which patients might be most at risk for this, and of course, eventually, perhaps some new strategies towards targeting. So if I take a step back and ask, what are we doing? The answer is, we believe, and we've devoted my career to trying to take ideas like this and methods from physics and use it to unravel complex systems, and in particular, over the last couple of decades, complex biological processes. This is something now that is almost accepted in many areas of simpler cell biology. If one looks at some work on bacteria, if one looks at work on yeast, if one looks at work on simple eukaryotic cells, then theorists are working hand in hand with experimentalists to try to combine these computational and simulational strategies with targeted experiments to try to create a new quantitative version of our understanding of simpler biological systems. And the basic hypothesis that we're devoted towards pursuing is that it's time to do the same in cancer biology. It's time to harness the power of theoretical physics to look through complex systems, identify fundamental mechanisms, and thereby work together with the cancer biology community to better understand the mechanisms, of course, all in service of creating better prognosis, better treatment, etc. All this, of course, requires a team, and I wanted to mention that, this is work on the motility aspects that I started before getting here, but what we've managed to do since coming to the Houston area, with the help of many people over at the medical center who have been extremely gracious in sharing their time and expertise with our theoretical group, is establish a core set of people over at Rice that try to apply physics ideas, modeling ideas to these questions in cancer biology. And I hope that we will be sufficiently successful that you will hear a lot from us in the years to come. Thank you very much.
That was beautiful, and I'm always excited by studies talking about studies that are quantifiable. And I think that's the approach you're taking. My question is very basic, and I may not even understand why I'm asking it. But what about the relationship of the matrix, of the milieu in which these cells find themselves? I mean, you seem to be concentrating on the cells themselves.
Right.
But there are signals that can come from that matrix. And we know that in the stem cell area, when there are caches and there's tissue-specific caches, that does have influence. So could you address that?
Yeah, so I guess we have to start somewhere, and I guess we started with maybe too overly cell-centric view of the system. We certainly recognize, even in the traditional stem cell literature, there are clearly works that show that, for example, even the mechanical nature of the environment can bias these circuits. So our view is, first of all, I tried to pick the simplest example. We do have work on going beyond how single cells make these decisions to what we think of as collective cell decisions, where decisions involve cells secreting factors. We've looked at the Notch pathway, we've looked at other pathways, and tried to understand to what extent cells are making individual decisions versus correlated decisions. So on the level of chemical communication between cells, we're actually working on that already. To the extent that the mechanical interactions with some of the extracellular materials makes a difference, that's actually a much harder problem, even from the physics end. It turns out that elasticity and mechanics is harder, even for physicists, than chemical communication, but we're just starting to work on that also. So I think the goal eventually is to understand, in some way, the relative importance of all those different factors in different contexts. One could also ask about when we're going to get around to thinking about the immune system. Yes. No answer. But I think I wanted to, first of all, present the sort of simplest possible story we had, because even that simplest possible story already can try to make sense of some confusing pieces of the cancer biology literature, and then we try to move upwards and forward towards including more of the actual complex nature of what's going on. So yes, I agree, but we wanted to start somewhere. Yes.
So this might be related to the previous questions, but as most people here know, tumors tend to metastasize to bone more than any other organ. So yeah, tumors tend to metastasize to bone, so producing metastatic bone disease. So have you looked at the characteristics of this circuit in terms of why they choose this tissue? Is it because of the physical characteristics, rigidity?
Yeah, so if I just repeat your question, you're interested in, for example, breast cancer or whatever, that have, and in general, we understand that there are there are organ-specific biases that presumably emerge from some match between the nature of the cells that are undergoing metastases and the local microenvironment. So I think that, I don't have anything specific to say about that. I think there is no yet quantitative explanation for any of that. From my perspective, it's one, I mean, so the answer is yes, we know that as a fact. I think that this field will have to try to come to grips with that as we move forward. So it's an unsatisfactory answer, but yeah.
Hey, I had a great talk. I had a question about subcellular localization. There's a lot of uncertainty about how biomolecules are localized inside of cells,
Yes.
and presumably that would impact how the terms in these sorts of models are coupled to one another. And I was wondering whether your work has made sort of can help guide future experimental work in terms of highlighting what types of structural information would be most valuable from the standpoint of quantitative modeling, and then what type is sort of, you know, kind of interesting but not useful from the modeling standpoint.
So I would say we've done that, but not yet in the context of cancer. So in other contexts, other signaling systems, again, in simpler cells, the community in general, and also me in particular, have looked at issues such as to what extent cellular compartmentalization of different pathways can make quantitative changes in how the cell is doing this information processing. So there are results about that. They have not yet been, so those results, again, I mean, the problem with a talk like this is a great field because every time you think of something, there's always clear what you also should be thinking about. So that has been worked out to some extent in some simpler cases. And they're known to be compartments in metabolism. There are known to be compartments in calcium handling. That has not yet been folded into these new, this cancer EMT model 1.0, let's say.
Thanks very much.
One very fast. Yes.
Thank you very much for a nice presentation, very interesting results. I'm wondering, in the models that you generate, obviously there is uncertainty, model uncertainty. So I would like to have your thoughts about what approach you use so as to introduce the uncertainty of the modeling component explicitly into the predictive element, as well as on the fact that the different actually cells are three-dimensional entities that navigate not by themselves, but within a cluster.
Well, I'll try just to offer a short answer. I mean, when we convince ourselves that some behavior, when we convince ourselves that some behavior is predicted by the model, what we actually test is, you know, is this really a robust prediction in terms of, is the structure of the network and the form of the interaction sufficiently specifically enables that, let's say, different phases to exist independent of uncertainties in the parameters. So if you want to predict what will the cell's response be to a given, you know, 2 milliliter bolus of TGF-beta under this condition, uncertainties in the parameters kill you. If you're interested in knowing what are the general types of states that can exist, what directions will transitions take place from one to the other, and what you might do to control the network as a general sense, then you can live with a lot more uncertainties in the parameters. So we use pretty traditional methods of just varying parameters, looking for robustness, but at least we try to focus on predictions that survive under those variations, because there are huge uncertainties. As the more complex you get the system, the more the uncertainties grow in terms of the fundamental parameters of the system. But yes, we try to do that as best as we can. Thank you.